Natural selection involves the following. So if you get a long question, maybe 7-mark question on natural selection, these are all the steps. First step, there's going to be variation within a population. This is caused by mutations or changes in DNA and the crossing over and independent assortment in meiosis. This video is going to involve meiosis because it's a topic that comes up frequently and it's very, it's kind of hard compared to the other topics. It's always good to revise it. Second step, there's an overproduction of offspring reaching carrying capacity. This is the point at which there's so many offspring, there's so many in a population that it just can't grow anymore due to lack of resources or space, etc. Third step, environmental selective pressures such as density dependent factors. Okay, so these are factors that depend on how big the population is. For example, intraspecific competition. When there's a lot of individuals in a population, there's going to be more intraspecific or competition within this population, if that makes sense. Density independent factors, they tend to be abiotic, so non-living. For example, climate. Okay, so it doesn't matter how big the population is, a hurricane is still gonna affect it the same way. Okay, so that's why it's independent of the density of the population. All of this links to another all other topics. Okay, so if you don't understand them, um, too bad. Fourth, survival of the fittest. So because there's a lot of competition and there's external factors, those with the right alleles, the right genes, the advantageous traits are going to survive. Okay, and then these are going to reproduce and pass on these alleles to offspring. Okay, so the offspring are going to inherit these advantageous alleles, and then eventually speciation is going to happen, which I'll also explain in a different video. So this is speciation is essentially the formation of new species. So this would be a very detailed answer of natural selection. Let's have a look at meiosis. I've got some animations cooked up for that, so get ready. Meiosis transfers diploid to haploid. Okay, so n is the number of chromosomes, so 2 times n becomes n haploid. So it halves essentially. So because the number of chromosomes halves from diploid to haploid from 2n to n, this is called a reduction division. Now, DNA replication must happen before meiosis. Let's have a look at the first phase of meiosis, which is prophase 1, and here's where the animations kick in. First thing that's going to happen, chromatin condenses into chromosomes, okay, and the nuclear membrane disintegrates, as you're going to see here on the left. Boom, nuclear membrane disintegrates. Now, these chromosomes are the following. They're going to be the joining of two sister chromatids, sister for identical, so they have the same alleles, they have the same genes. So these are going to form what's called a bivalent. So this is a bivalent, bi for two, okay, held together by a centromere. It's this circle in the middle, this black circle is a centromere. This whole thing is a bivalent of two sister chromatids joined together. And now, before anything else, crossing over is going to happen. As you can see here, these sister chromatids are identical because they're, well, sister chromatids. Red one and red one and blue one and blue one, they are identical. But they will no longer be identical because between different chromosomes, there's going to be an exchange called crossing over. And this exchange involves the exchange of DNA segments, DNA fragments, okay? So these swap, and because of that, well, first they swap by the chiasma, and because of that, the sister chromatids are no longer genetically identical to one another, and they become what's called recombinant chromatids. Now, the chromatids that are not involved in crossing over and are still genetically identical are what we call non-recombinant chromatids. So make sure you note that down as well. And finally, we can see that gametes, so the sperm cell and the egg cell, inherit different combinations of alleles from the maternal, from the mother, and paternal, sorry, chromosomes. And this process is thanks to crossing over during prophase 1 of meiosis. Back in prophase 1, we can see that centrosomes or microtubule organizing centers produce bundles of microtubules called spindle fibers. So these centrosomes or MTCOs produce these spindle fibers right here, this purple thing. And centrosomes are for animal cells, and plants involve microtubule organizing centers or MTCOs, and these both have the same function, which is producing the bundles of microtubules called spindle fibers. And have in mind that centrosomes contain something called centrioles. At the end of prophase 1, this is what the cell is going to look like, more or less. Okay, so the sister chromatids, well, they are no longer sister chromatids. The recombinant chromatids are going to be around the center, okay, of the spindle fibers. The centrosomes or MTCOs are positioned to the sides. And now we move on to the metaphase 1, the second phase of meiosis 1. Okay, here the spindle fibers are going to attach to the centromeres. So the purple thing is going to attach to the black circles of homologous chromosomes. So now we call them homologous chromosomes. Okay, so as you can see, they attach to the purple thing. So they're lined up by the equator of the cell. You know, Earth, there's the equators. It's a similar shape. Anyways, the maternal and paternal homologs show random orientation towards the poles. This is very important. This links to natural selection about how genetic diversity is formed. So this also happens in the metaphase 2 of meiosis 2, further increasing genetic diversity. And this is called independent assortment, where the positions of maternal to paternal and maternal to maternal chromosomes are independent of one another. Random orientation. Keywords linked to natural selection. Now, anaphase 1, after all this random orientation, independent assortment, and lining up by the equator of the cell, we have that the following is going to happen, okay? These spindle fibers have shortened, as you've just seen, and they have separated the bivalent or tetrad. Now, these are going to pull homologous chromosomes apart and towards opposite poles. Okay, so these two have been pulled to the right pole, these two have been pulled to the left pole, and this is the difference. Moving on to telophase 1, the cell has elongated, as you just seen, and the homologous chromosomes reach the poles and decondensed. Okay, I couldn't represent how they decondensed, couldn't find an animation, but essentially they've reached the poles, the ends of the cell after it has elongated, and the spindle fibers have pulled them here. Now, these spindle fibers are actually going to break down into phase 1, as you just seen, and a nuclear membrane that disintegrated in prophase 1 is going to form again around each nucleus of the new two cells we're going to have the division. But before that, there's interkinesis, which is the period of rest between meiosis 1 and meiosis 2. Moving on to meiosis 2 now, after cytokinesis. Cytokinesis 1, it's simply the division of these. Okay, so before the cells used to have 4 of these homologous chromosomes, and now the cells have two. So the number of chromosomes halved, and this is why meiosis is a reduction division. As always, any questions can go in the comments. Prophase 2. Okay, so we're starting with meiosis 2. What's going to happen? The nuclear membrane disintegrates in both cells. The homologous chromosomes recondense. Again, I couldn't show an animation for this. I'm sorry about that. Just remember that. And empty cells migrate to opposite poles, as you've just seen right now. Spindle fibers start to form. Okay, not fully. They fully form by metaphase 2. Perfect. Now these spindle fibers attach to the centrosomes, as you've seen here, the centrosomes of these homologous chromosomes, and they show random orientation towards the pole. The poles, remember, this independent assortment. Again, this further increases genetic diversity. Anaphase 2, what happens? Oh, the spindle fibers have shortened, as you see in these purple things have stretched to the sides. They shortened. They split the centromere, so you can see the black circle isn't here anymore. It's split these in two, and it's pulled the sister chromatids, well, not necessarily sister chromatids, the recombinant chromosomes, apart towards the opposite poles of the cells, the opposite ends. And now once these are separated, they're called chromosomes. Oh shit. Anyways, telophase 2. Again, the cells elongate. Well, now it's cells. Before it used to be cell in telophase 1. The chromosomes reach the opposite poles of the cells and decondensed, and the nuclear membrane forms around each nuclei. What happens next? You guessed it. Cytokinesis 2. Well, you don't necessarily need to call it cytokinesis 1 or cytokinesis 2. It would just be cytokinesis for both cases. So it's going to happen. Boom. 4 cells. We went from diploid to haploid. And again, the number of chromosomes has divided. It went from 8 chromosomes to 4 chromosomes, and now 2 chromosomes per cell. Wow. 4 haploid daughter cells is the final product of this division. We're on to the actual ecosystems topic. Biological fitness. Okay, this is an organism's ability, fitness, to survive, reproduce, pass on its offspring, blah blah blah. So this involves traits that help it survive or to reproduce, camouflage, resistance to diseases, or traits that help it attract mates or produce more viable offspring. Bright feathers, effective parenting, aesthetics, muscles. Um, Interspecific competition, which I've mentioned before, is intra, it's within the individuals of the same species, and it drives natural selection. Okay, interspecific is among different species. Now, only heritable traits drive evolution, only the heritable alleles are going to cause the natural selection, the speciation, and the acquired traits do not affect offspring or evolutionary change. So acquired traits are not the same as inherited traits. Acquired traits is, for example, let's look at an example. So heritable traits, these are genetic or DNA based, and they are heritable, of course. And examples of these are eye color, you get it from your parents, or the pattern of fur from parents. Now, acquired traits are traits that are caused by environmental or lifestyle factors. They're not genetically inherited. So if you go to the gym and you make muscle, you don't get that muscle from your parents. You got it because you went to the gym, right? So that's why it's acquired. It's not heritable. An animal example would be flamingo feathers, okay? And they get these from their diet. Basically, they are not inherited. Sexual selection, if you do psychology, you'll know what this is, right? A special type of natural selection that is just involves finding a mate and reproducing. Okay, there's two types. There's intrasexual, intra within the population. Okay, so this is males competing with other males. Well, actually, inter is actually within the population as well, but in a different way. Intra as in the same gender. Okay, so males compete with other males. This involves strength, weapons, large size, fighting each other. Examples: deer antlers fight each other, and elephant seals. Okay. Now, intersexual, different genders, females choosing the males. Okay. So they'll choose based on, for example, colorful feathers, plumage, the songs that they sing, if it's birds. Okay. And yeah, examples are birds. When we're talking about ecosystem stability, there's two main ones that come to mind, which are fairly stable: tropical rainforests and coral reefs. Why are these stable? Well, they have a lot of resistance, okay? Despite all the anthropogenic disturbance, they're still up and running, though in the future this will not be the case. Ability to recover after disturbance. If we leave them alone, they'll probably recover pretty well unless we hit tipping points, okay, points of no return. Uh, they have good supplies of energy from the sunlight, nutrients in the soil. There's a lot of carbon and nitrogen cycling. I believe this is a different topic. Good climate, water availability, topography, and they have a lot of species diversity. There's a lot of different species, a lot of different habitats. You know, rainforests have rivers as well as the trees, and genetic diversity. So the species within have a lot of mutations. There's a lot of speciation and natural selection going on. Now, we can actually model ecosystems into simpler representations to understand the whole thing. So we can make a mini rainforest, mini coral reef, okay? So this is called modeling ecosystems. We use them to identify tipping points or points of no return when changes become irreversible. The damage is going to be there forever and you can't do anything to fix it anymore, okay? So an example of these are mesocosms, okay? So let me underline you. They're useful for studying effects of climate change, nutrient levels, or pollutants. Okay, so add a bit of pollutant to one of these little mesocosms and see what happens. Okay, and it represents what happens in the larger scale. Now this can be outdoor or indoor. These inside the glasses would be indoor, of course. They work best for aquatic or microbial ecosystems. Yes, I agree with that. And there's a lot of variables to have in mind. The pH of the water if it's aquatic, temperature, light intensity, the color. Okay, I guess that would be like the color of the glass that it's contained in, or the color of the rocks, etc., the abiotic factors, maybe even the biotic ones as well. Concentration of ions or nutrients, and the size of the population of different species. Now, there's going to be some energy exchanges. Okay, so through the glass, we know that sunlight can enter, right? And this means that even if the plants are inside, the aquatic plants or the non-aquatic plants, they can still photosynthesize through the glass, okay? There is less sunlight because the glass filters some of it, but in the end they can still photosynthesize, okay? And they're small plants, so it's not like they need much anyway. Now, energy can be exchanged, but matter cannot be exchanged with the outside of the mesocosm. Okay, so the water, the carbon, and the oxygen cycle continue internally. So water, carbon, oxygen, it doesn't leave the mesocosm, it doesn't leave the jar, but it keeps recycling inside. Okay? Now, sustainability is achieved is these biotic and abiotic factors, biotic being living and abiotic non-living. If these are balanced, you can continue internally. You can keep going, and your mesocosm can carry on for like maybe forever, really. And as I said before, because the energy can flow inside and can produce, the plants can photosynthesize, but the matter cannot be exchanged with the outside. This is what we call a closed system, which is actually linking to another topic in this IB biology course. You also need to understand causes of unsustainable agriculture, farmers and stuff. I'm gonna use an example as well, eutrophication. We're gonna look at it in a second. So it's gonna be generally caused by agrochemicals, fertilizers, pesticides, overuse of water to grow the crops, soil erosion, uh, stealing a lot of nutrients, occupying a lot of space, so biodiversity loss, and carbon footprint, okay, from the animals farting and releasing methane. Anyways, so there's going to be some principles, the where am I drawing? The IUCN principles, okay? They're going to ensure or try to ensure proper harvesting of crops, okay? So, first one, maintain populations above a minimum size, avoid depletion. You don't need to know this exactly, but it's good to look at it once, or twice, just in case you get a multiple choice question on this. Ecosystem-wide concern. Okay, so policies should protect all renewable resources, not just one species. Okay, pretty straightforward. Uh, the ecosystem must be maintained to prevent irreversible change and minimize long-term damage. And you should always monitor and research. Okay, pretty basic. Uh, I don't even think you would get a long answer question on this. It would just be multiple choice. Sustainable harvesting would be when you remove the maximum yield without decreasing the ecosystem stability. Of course, we want maximum yield. We want as many crops as possible, but at the same time, we don't want to affect ecosystem stability negatively. Now, there's a keyword that you do need to know 100%. I'm circling it right now. Keystone species. Let me change color on board of the red. So these cause cascades or chain reactions, chain cascades of events through the food webs. Okay. So for example, a keystone species in the river could be the beaver because the beaver builds dams. Okay. And these dams are where a lot of other species can live. Okay. It creates a whole new ecosystem. So without the beavers, as an example, this ecosystem wouldn't even exist. It would collapse. Or nitrogen fixing bacteria. Without these bacteria, there would be no nitrogen cycle, I believe, and because of that, the ecosystem just could not exist. So they are very important. Another example would be bees. You might have heard bees are extremely important, and if they were gone, we would all be cooked and die. The ecosystem would be dramatically altered or collapse without these keystone species. Eutrophication, okay. Oh, what's that arrow there? Should have an animation. So what happens first? Farmers are going to use fertilizers. You might have done this at GCSE. These fertilizers contain nutrients such as nitrogen and phosphorus, and they travel around soil, they travel around the air, and again, eventually they get to water bodies, to lakes, rivers, etc. So these now have excessive nutrients, right? So these first fertilizers, sorry, are going to reach these water bodies, and so the algal is going to bloom. It's going to grow a lot because it has so many nutrients more than it's used to. And because of that, because a lot of algae is growing, a lot of algae is going to die, and that means that a lot of dead organic matter accumulates at the bottom of these rivers, these lakes, etc. Now, this dead organic matter is decomposed by bacteria. There's more of it, so more bacteria decomposing, and when they decompose, they're going to carry out aerobic respiration. Okay, so there's going to be an increase in biochemical oxygen demand. So there's going to be less in the water because these bacteria are going to consume a lot of it through their aerobic respiration. And lastly, because there's less oxygen in the water, marine animals die unless they're adapted to these low oxygen levels, such as certain species of turtles. Two keywords right here. So we have bioaccumulation, which is the accumulation of pollutants within the tissues of organisms. For example, microplastics, which are actually non-biodegradable. This means that in nature they cannot degrade, they cannot break down naturally. A plastic water bottle can be in nature for millions of years and not degrade, okay? Second keyword, biomagnification. So this is the accumulation and magnification of pollutant concentrations through the trophic levels of food chains. Okay, so the more is accumulated through, so how to explain this? Through the trophic levels, for example, there's animal 1 and it's eaten by animal 2, and this one is eaten by animal 3, and every single time there's more and more microplastics involved. Okay, so when you get to 3, there's a lot of microplastics. And so the effect is magnified through the food chain. Okay, 1 accumulates plastics, 2 is also accumulating plastics, and not only is 2 accumulating plastics, but then eats 1 and has even more plastics. And then 3 is accumulating plastics at the same time and then eats 2, and there's a lot of microplastics in the end. So it's accumulating for every single one, and through the trophic levels, it is magnifying, it is increasing, if that makes sense. Okay, any questions can go in the comments. But anyways, carrying on. Rewilding. Okay, this is something separate. Rewilding. How do you rewild after doing damage? This is the case where you haven't hit a tipping point, okay? So different things, reintroducing species, especially keystone species, restoring the habitat by afforestation, adding trees and removing the invasive species, okay, like the alien species that aren't supposed to be there. Links to another topic. Rewilding the urban areas, that's why parks exist, introducing nature into cities, rewilding rivers and waterways, removing barriers like dams. This is basically common sense that you might use in a question, but just look at it once just in case. And who cares about this? Almost finished. I've seen a 6-mark question on this, so watch out. Anthropogenic or human-caused climate change, okay? Um, you've probably heard of greenhouse gases, CO2, CH4, methane, and nitrous oxide, N2O. Positive feedback cycles, okay? Positive, not in the sense that they're good, in the sense that they just keep getting increased and worse over time, okay? So the more the cycle happens, the more it happens, if that makes sense. It just exponentially increase. Anyways, 3 examples that you definitely need to know because I have seen a 6-mark question on this and it can come up again. Who knows? Now, I have a snowflake here to help you remember the albedo effect, which is that when ice melts, there's less reflective surfaces, and so more heat is absorbed, reflecting the light, okay? So it reflects the light, and if there's less of it, it reflects less light, and there's more heat absorbed into the surface of the Earth. When there's more heat, the ice melts faster. And so what happens? If the ice melts faster, there's even less reflective surfaces, and this cycle just keeps on going and going and going and getting worse. Okay, any questions again? Go in the comments. Next one. I'm changing color now. You. Warmer temperatures increase the rate of microbial anaerobic fermentation, respiration. Okay, and this releases more CO2 and methane, greenhouse gases. There's a bacteria here to help you remember. Okay, and if there's more of these gases, there's more climate change because they're greenhouse gases. And if there's more climate change, the temperatures increase more, and this increases the rate of microbial anaerobic fermentation even more. Okay, get where I'm going? Last one, remember this one as well. When there's more forest fires, okay, there's more deforestation. Logical, right? And this decreases how much carbon sequestration there is. And so there's more carbon in the atmosphere. If there's less trees, there's less carbon sequestration. They don't use the carbon in their photosynthesis. And so more climate change, which increases deforestation even more because the warmer temperatures lead to forest fires. Boom, simple as that. Ocean currents, two types. The ones in the surface of the ocean, which are driven by wind, and the ones in the deep ocean. Okay, at the bottom of the ocean, they're driven by water density differences, temperature, and salinity. Now, the melting of ice and glaciers add fresh water, and this fresh water reduces the salinity and alters water density, increasing the number two, deep ocean currents. Okay, and linking to surface currents, the first one, wind patterns can shift the current direction and strength. And these disruptions affect heat and nutrient distribution. Essentially, they affect ecosystems, weather patterns. Okay. Now, another thing to have in mind on how these surface currents matter is that deep nutrient-rich water rises to the surface, supports organisms like phytoplankton. Now, wind-driven upwelling occurs when wind pushes the surface water away from coasts. Okay. Now, this upwelling, again, gives the phytoplankton the nutrients they need. Okay. And the warmer surface water reduces this upwelling. And this limits the nutrients that these phytoplankton can get, threatening marine food webs. And so, because of this, we can deduce that the wind is good for the phytoplankton. It gives them more upwelling of nutrients, while the warmer surface water, the hotter water, is bad. Have in mind, species move to higher latitudes, poleward, or elevations upslope to find suitable climates. Basically, they move to find better climates. And the drivers of this are the rising temperatures from climate change and shifting precipitation or climate patterns, okay, weather patterns. And now in coral reefs, there's a bunch of different threats, okay? First one, ocean acidification caused by the excess atmospheric CO2 that dissolves into seawater. Okay, this isn't chemistry, so you don't necessarily need to know this, but just understand that CO2 mixes with the water, the H2O. Okay, this is a reversible reaction, but well, that's irrelevant. Um, this forms, I believe, carbonates or something like that. This carbonate turns into hydrogen ions. Okay, and another thing, so when there's a lot of hydrogen ions, this lowers the pH of the water and makes it more acidic. Okay, and because the water is more acidic, this reduces something called chloral calcification. Let me underline that. Coral calcification. So lower pH, more acidic water. And this weakens the reef structures, okay, affecting the ecosystem. Then we have coral bleaching. This is from the rising temperatures of the water or pollution. So corals expel something called zooxanthellae or symbiotic algae. So they lose their color and energy sources, leads to weakened corals and potential death. So these are the two main threats to coral reefs. Carbon sequestration, key definition here. So this is capturing and storing CO2, sequestration in Spanish, secuestro. links to that, to mitigate or stop climate change, okay? Two different things. Afforestation. This is a form of carbon sequestration. Planting trees on non-forested land, so land without forests. These trees absorb CO2 via photosynthesis, storing carbon in their biomass, okay? Leaves, trunks, and roots, and these can be large scale or small scale. Restoration of peat-forming wetlands. So peatlands store large amounts of carbon in waterlogged soils, so soils full of water and, well, an overwhelming amount of water. And human drainage for agriculture releases this carbon. So this is another form of carbon sequestration we can do.